Hi, I'm Jan Witkowski. I'm Director of the Banbury Centre here at Cold Spring Harbour Laboratory. And this is lunchtime on the 5th day of the 79th Cold Spring Harbour Laboratory Symposium on Quantitative Biology. One day we're going to have to shorten that, it gets too long to say. And the topic of the symposium this year is cognition. And I'm very pleased to have Carl Deisseroth with me from Stanford. Carl is doing some of the most exciting experimental work in neuroscience. And you're speaking this evening, Carl, so
that's right.
How about giving us a little foretaste of what you're going to do? You're going to talk about two topics, optogenetics and clarity.
That's right. We've been working on developing optical technologies for exchanging information with the brain of experimental animals that are carrying out complex behaviors, and optogenetics is one of those. It's a way of using light to control well-defined sets of cells within neural circuitry during behaviors, turn them on or turn them off, and assess the causal impact on behavior. And what I'll talk about tonight is a social behavior, and that's, social interaction is, in many ways, it's a very high-level cognitive task. It's a difficult task. That's one thing, but it also carries risks, and an organism has to be motivated to carry out a social interaction. And so we'll present some results on optogenetic control of circuits that turn up or down the tendency to engage in social interaction, at least at the level of mouse behavior.
So what sort of social interactions are you looking at in this experimental setting?
Well, a lot of great work in the past has been done on courtship and cross-sex affiliative behaviors. We're taking a slightly different tack, which is same-sex conspecifics, mice of the same species, their willingness to or desire to simply be in the same location as each other, and they have a natural tendency to do that. So if you give animals a choice of being alone, or with a novel object that's of some interest, or with another mouse, same species, same gender, they'll tend to prefer to be with the mouse of the same species, and there'll be active engagement and approach, and it's an interesting built-in process that is present. And so we're interested in what drives that, what turns that up, what turns that down, and so we quantify the amount of time the animals choose to spend engaged in this behavior.
And what's known about what neural circuits or neurons, brain areas, or what's already known about what's involved in this behavior?
Well, there are these different aspects of social behavior. It is a challenge, but it also requires motivation, and those two aspects map on to the neural circuits that seem to be involved. The prefrontal cortex, medial prefrontal cortex, that integrates much information and helps generate plans and move toward goals, is certainly involved in this sort of behavior, but also deeper subcortical structures that involve recruitment of reward and motivation pathways are also involved. And so I'll talk about those different aspects and how they play into this behavior.
So this almost raises, I was going to say, sort of not frank, so almost robot thing. So do you think that using optogenetics, and by manipulating particular neurons that may be involved in these processes, you'll be able to drive the mice to particular types of behavior?
Yeah, I mean, this is a deep question. We're playing in patterns of activity. We're turning up or down certain kinds of behavior, and so yes, the short answer to the question is the mice can be modulated in their behavioral choices, and even the kinds of behavioral choices that they would carry out. At the same time, we don't want to just play in activity that we think is relevant. We'd also like to see what the animal is normally using. And so another thing I'll talk about tonight is a new technique called fiber photometry, which lets us collect information on what the animal is normally using in the course of its social decisions and social choices. And that's a very powerful thing, because then we can see what's normally present and try to mimic it or provide it in our experimental interventions.
Can you say very briefly about how this, did you go fiber photometry?
Yes, fiber photometry, and it's a trick that uses a optical chopper to break the incoming signal at a very specific frequency, and then we have a detector to pick up signals coming out of the animal that's also tuned to detect signals at just that frequency. The advantage of this is this lets us detect very small signals that otherwise would be buried in the noise, because only the signal we're interested in is modulated at this very strange frequency. And so it's a trick that lets us, even in a mouse that's freely behaving in a well-lit room, we can pick up activity signals with fluorescent indicators of neural activity that are buried deep within the brain in some of these deep neural circuits that are involved in these social behaviors.
So these are free roaming mice.
Yes.
So this. Are there implanted things in their heads to detect these signals?
There's a fiber optic, a single fiber optic, and a single thin fiber optic that is able to implement either the optogenetic control or the fiber photometry detection of the signal. In both cases, and this is the key advantage, same system, same surgery, same location, same site, one thin optical fiber, both delivers the control and can pick up the activity signal. And so then in the same animal, same preparation, we can get both data streams.
And do they use the same fluorescent compounds to...
Well, for the control, we use these proteins that are encoded by microbial opsin genes, and these are genes that come from algae, from bacteria. They respond to light and open ion channels in the membrane. So they themselves don't generate an optical signal, but they use light to move ions across the membrane of cells that we're controlling. Then we pick up the signals, we detect and observe the native activity patterns using a different protein, and these are genetically encoded fluorescent reporters of ion flow. So two parallel sets of proteins that help us do the job.
And these are transgenic mice. How have you introduced these?
In both cases, we inject a virus that carries the gene encoding for these proteins. So we take mice that could be transgenic, but they also can be wild-type mice that have no label.
But essentially, it's a virus that's used to introduce these,
That's right.
These molecules.
That's right.
Yeah. I was just thinking about Eric Nestle gave a very interesting talk about his model of depression in mice, and you can quite easily see that he has mice that are susceptible to depression, the way that he induces the depression state, mice that are resistant, and he has sort of proper controls. Quite easily see how your sort of system could be used to interrogate what's actually happening in the brains of these mice.
Yes, in fact, it's funny you say that, but Eric, who's done some great work in this domain, and we've actually collaborated on some prior work, he's quite interested in applying the fiber photometry method to his paradigm.
Yeah. I'm ahead of the curve for once.
Yes.
Okay, so what's the current state of clarity at the moment?
Yes.
Very clear. Yes.
Getting clearer.
Sorry.
One of the press pieces had a nice quote. They said, "I can see clearly now the brain is gone." And what we do with clarity is we are able to use a chemical engineering trick to make the brain transparent, and it looks like it's gone. It's quite a remarkable thing, but it's not. All the scattering, the light scattering is gone, and that lets you see through the brain. We do this by removing the lipids, removing the fats, and before we do that, we make the brain robust to such a dramatic thing by building in place a hydrogel which keeps all the proteins and cells in place. And we published that about a year ago, and the first papers using it are starting to come out. A nice one in spinal cord just came out from another group just a month or so ago. But one thing we've been working on is trying to make it faster and easier to use, and so we have a paper coming out later this month, in which I'll talk about on making the whole process and the data acquisition faster by orders of magnitude, combining clarity with light sheet microscopy, and we call this clarity optimized light sheet microscopy, and it lets you collect volumes 100 or 1,000 times faster than before.
How long does it take to prepare a brain as you were doing it, say 6 months or 9 months ago?
For a mouse brain, the actual preparation process takes between a couple days to a couple weeks, depending on how much you want to stain for, label for. But the whole process of actually making the brain transparent and creating the signal is a couple weeks. And then what we've really sped up is the imaging process. Whole brain imaging now can be done, and instead of involving days, it's now in the matter of tens of minutes or hours.
And this is you imaging in what technique?
Yeah, so it's a very interesting kind of technique. It's a 100-year-old concept called light sheet, and what you do with light sheet is you scan a plane of light through your tissue, So instead of scanning a point, which confocal and two-photon microscopy use, very nice high-resolution methodology, but it's quite slow. And they also have the disadvantage that you're imaging at a point, but you're still illuminating your whole sample with light with confocal and two-photon, which creates bleaching problems and degradation of your signal. With light sheet, you're only illuminating where you're also imaging, and so it's very efficient in that regard, and so you're not destroying your signal in other parts of the tissue. You're illuminating where you're imaging, and you're scanning a plane, and it's much faster. Now, it required some adaptations to make it well-suited to these sort of intact tissue preparations for the clarity treated brain, or clarified brain, as we say, and that is what we're going to present and talk about tonight.
So what's the I guess thickness of these sheets as you're going through?
Traditional resolution in that axis has been considered to be limited to the sort of 10 to 20 micron range, and that has to do with the thickness of the light sheet. So exactly what you're asking. But this is something that can be improved upon with certain kinds of innovations, which we'll talk about. And so we can get now, even deep within the tissue, a micron level resolution. We can see fine processes, axons and dendrites, even deep in the middle of the mouse brain. And so that's an exciting advance. Not only do we have the speed, but we can maintain this high resolution.
So, these, of course, are fixed preparations.
Yes.
So what sort? Of course, what we would like to do is get some insight into dynamics. So, do you have a mouse that's been trained to do something or respond in some particular way, and you compare that brain with the brain of a control mouse? What sort of questions can you ask using this technique?
Well, what we've been working on is making sure that you can do all of this in the same mouse, not at the same time, but the same mouse at different times. And this is what this integration of data streams is a very powerful thing, as you indicate. So you can now have a mouse that you've done an optogenetic control experiment on. You've turned on or off different kinds of cells. You know for that animal its behavioral score. You might even in that same animal have done a fiber photometry experiment or other kinds of imaging to see the natural patterns of activity in that same mouse during that same behavior. Then what you can do now is take that same brain and you can put it through the clarity process, and you can register or align the data sets you get, even though very different times, different conditions now outside the animal, but you can then register the data sets and say, okay, this region and these cells, well, these were the cells that were doing such and such during behavior. Here's their phenotype, here's their wiring, and so that the goal is to move toward a more integrative understanding of the neural circuitry.
Yeah, that's fantastic. It occurs to me, how generally applicable is the clarification technique? Have people tried it with other tissues, other organs? And so
Yes,
on?
that's been an explosion of interest. So people have sent us pictures of clarified hearts, pancreata, even whole embryos. And so it's people are and there's subtle tweaks to each sort of preparation that are needed. For example, one thing we found in the mouse brain, we have traditionally, as we published, that to speed up the process, you can accelerate the clearance of the tissue by imposing an electric field, and you can accelerate the clearance of these lipid-containing micelles out of the tissue. Well, what we're finding is that for the brain, you usually don't have to do that. Actually, you can do what we call passive clearing, and it's fast enough and good enough, and you don't even need the electrophoretic process, which has been a boon for a lot of people who didn't want to get the whole electrophoretic process going. But what people are finding for other organs, the heart, the electrophoretic method is very important. You can't dispense with that. You've got to have and that has to do with tissue properties, and there's different things for every tissue, but we're seeing it applied across biology.
It'd be fascinating to apply it to embryos.
Yes.
I was just thinking back when I was in I guess it was a German company made these wax models of embryos, and these three-dimensional wax models, and to sort of be able to see that sort of structure in the real embryo.
Yeah.
Fascinating.
I actually got my start in science as a developmental neurobiologist. Those are my first questions that I cared most about, and so it's been exciting to see these clarified mouse embryos now.
You know, I can remember when we were talking about this, I guess when you were teaching on a course here in the summer, you were saying that it was easy to lose these brains, these
Yes.
clear brains. Is that still the case?
Well, the hard part is representing them in the published page. That is the difficult part because there's nothing really to see, and so how do you show a picture of that? And so we've been trying different strategies, putting the brains over text, over grids that you can read through, and so on. But in the end, people mostly care about the data, and so that's what we focus on presenting now.
That's good. Carl, thank you very much.
Thank you very much.
Thank you.